Sono rimasto piacevolmente colpito dall'elenco dei nomi delle persone che hanno collaborato a questa provvisoria vittoria. In medicina le vittorie sono sempre provvisorie, per fortuna. E ho notato che hanno nominato 2 studenti. Ecco, e in più c'erano professori, ricercatori, ma 2 studenti che hanno partecipato all'impresa straordinaria. Io non capisco nulla di quello che dicono, non per l'invio dell'inglese anche, ma perché è talmente irta di scienza che io soltanto da stare lì a guardare come ascoltando l'aria di un'opera. E quindi sono emozionato nel dare subito il palcoscenico al professore Eduardo Fernández, che si occuperà dell'impianto corticale. Non so di che cosa si tratti, lo imparerò alla fine del suo discorso. Grazie.
E sarà capace di stupire te e tutto l'auditorium. Eduardo, prego, Università di Elche,
Integrazione
Alicante.
elegantissima, grazie.
Grazie mille per le presentazioni, Michael. Prima di tutto vorrei ringraziare il professor Andrea Cusumano e la Macula Genoma Foundation per l'invito a partecipare in questo incontro. È un grande onore e un piacere essere qui oggi, ma italiano è molto limitato, quindi mi scuso e parlare in inglese. So, ok. Okay, as you know, visual impairment is a huge problem. We have seen today that many people, we are talking about millions of people, has a visual impairment. And what happened in these patients, yesterday we were talking walking through the photo, and what happened on many of these patients, they are losing vision. And in many, or in some of these patients, at the end, there is no vision at all. So what happened? Okay, what can we do in this kind of patient? We are seeing in this meeting that there are 2 different approaches. They are not mutually exclusive, so it's one more biological and one technological approach. And in our lab, we are working in both. We have seen today that, for instance, drug delivery or gene delivery to the retina is a problem, because usually the eyes and the retina is isolated to the body. So traditional approach are rather limited. So Professor Girard showed, for instance, that they are working on a different approach using 2 different viruses for the Stargardt disease. And in our lab, we are trying different approach. We are exploring the different approach. For instance, instead of virus, because one of the problems with the virus is the size of the DNA they are able to release. But other problem, they are virus. That means that they can mutate. So we are trying to develop new formulation based on different kind of nanoparticles. And you can see here is for gene delivery to the retina. If we use subretinal injection, we got mainly the photoreceptors layer, or with intravitreal injection, we got mainly in inner retinal layers. And we can also use these nanoparticles for releasing optogenetic tools. Today morning, Professor Serge Picot showed there is some really amazing results with optogenetics, and we have very good results, not in human yet, but just in animals. You can see this is using just the crimson, and you can see the patch gland. Just the cells are able to have spikes and so on. And right now we are also working on different kind of nanoparticle. In this case, we are using even nanodiamonds. So with nanodiamonds in the nanoparticle, we are able to transfer many cells. This is in vitro, but we are improving our formulations. And in the lab, we are also using several animal models. We have transgenic and knockout mouse model. We can induce retinal lesion by laser, and we are also trying to develop new animal models, big models. And you can see here, we are using the same devices, clinical devices for animals. You can see this is an OCT, it's a standard technique in the clinical ophthalmology, and we are using the same device for mouse and for human. And we can follow the progression of the disease, or the progression of the new drugs and so on without killing the animals. And we are also developing, this is a pig model, for macular degeneration, the atrophic macular degeneration. You can see higher magnification. Where is it's not possible, the pointer is not working, but you can see the place, the location of the macular degeneration, and we can follow even different kind of treatments. But we have seen today also there are many diseases in which the retina is completely destroyed or damaged. There is a severe damage of the retina. They are in the image. You can see, for instance, an advanced glaucoma and diabetic retinopathy, optic neuritis, optic atrophy. So in these cases, has no sense to deliver one gene to the retina or to promote the regeneration or even to use a device at the retina, because the retina is too damaged. So we have to try a different approach. And in our case, we know that the higher part of the visual system is still perfectly functional in spite of the damage at the retina. So one of the ideas is to send information from the outside directly to the part of the brain who is processing visual information. And we are trying, in the lab, we are trying to demonstrate the feasibility of this approach. The idea is quite simple. Is as any other kind of neurological device, first of all, we have to collect the same kind of information that the system normally collects. If we were talking about one auditory problem, we have to collect auditory information. In our case, we have to collect visual information. Then we have to process that information in a more or less physiological way. And finally, we have to send that information to the right place. The right place could be the retina, but in some patients, for instance, with damage of the cable from the eyes to the brain, is not useful. So we are using a penetrating electrode at the layer 4. That is the place where the information from the LGN goes to the brain. And this is the idea. The electrodes are very close to the neurons. We try to stimulate, and this is the whole system. So right now we are trying to demonstrate the feasibility of this approach. And we are coordinating a clinical trial. In our case, is to demonstrate the feasibility to be able to provide to the blind people we call functional vision. It's not to recover the vision, because that is not possible right now. So for us, functional vision means for tasks like orientation, mobility, reading big characters in a screen or even numbers. So the question we are having ourselves are very simple. What kind of signals should we send to the brain? How stable are these perceptions on a day-to-day basis? Are the same today than next week or next month? How is the optimum space between the electrodes, and how many electrodes do we need to provide helpful visual information? And these are the clinical trial selection for the patients. So we are looking for patients with profound blindness, bilateral profound blindness, and no significant benefit from any conventional aid. So for us, it's also very, very important that to have a detailed medical history, including the documentation of the onset and evolution of the blindness, low risk associated with surgery, that means good general health, and it's very, very important that they fully understand that this is still research. because we are still at the research stage. This is not yet a treatment. And we are using this so-called UDAS electroarray. It's very, very tiny. You can see it's 4 by 4 millimeters, and there are 100 microneedles in this 4 by 4 microelectrode array. And one nice thing here, you can listen. This is the sound of the neurons. So we are able to see if the neurons are still alive, and we are able to see what happens when we send electrical stimuli. These are the spike information, but we also have kind of a micro EEG, is the local field potentials. And this is a typical experiment. Well, before. Okay, so right now we already have 4 blind volunteers, and we implant just at the occipital pole. And in the first 3, we know for sure that the implant location was at the primary visual cortex, mainly B1. In one case, some of the teeth were in B2. But our latest blind volunteer, Max, is he's 27 years old. He was finishing the law studies at the University of Kiev, and then after finishing, went to the war. It was a mine explosion, and he lost both eyes. Max has no eyes, but due to the explosion, he still has some metallic fragments in the head, so we were not allowed to make a MRI. So in this case, we use just a CT scan. You can see the location. We finished this in the same location, but it's impossible to know because we are not allowed to make an MRI. And this is a typical experiment. So the patient is sitting with the electrode, and we are seeing what happened in the brain, and then we just send one electrical pulse, and the patient is able to tell us about the description. It's a small walk, dot, and we ask about intensity and so on. So we have all the information in real time, and we can modulate, for instance, the brightness depending of the current or depending on the number of electrodes. We can also modulate the size. But one important thing, we were able to induce some patterns, pattern stimulation. And you can see here the pattern for the big letter O or the small letter O. And in question, in less than 1 week, she was able to fully differentiate the big letter O from the small letter O. And we asked many times, but what's exactly your perception? Because the problem, there is not any device able to tell us about the perception of what person. The person has to tell us, my perception is, and we ask many times, but is a letter or is something like looks like a letter? What exactly? And then the next day, she comes to the lab with 2 small sequences. You can see here, well, these 2 small sequences, and she told us the small one is the small letter O, and the big one is the big letter O. So it was clear that the idea we would try to induce. And this is the first time we have the whole device, and we switch on the device, and we ask where's the border between the black and white, and it was perfect. Nobody expected this, nobody in the team. So, and she said, okay, here is the, yes, it was there. So, but nevertheless, one of the problems with this kind of technology, I mean, it's very tiny, and this means that the field size, the perception is also in a very tiny place. And this is like, you have been in Alicante, this is the Esplanada of Alicante, it's like looking the Esplanada like this, of course no colors, in black and white, and then we have to discover what is outside just from this kind of information, very poor information. But nevertheless, the patients are able to use this information. You can see here another blind patient. The task was to find the mobile phone. It's moving the head, it's moving the eyes. It takes some time, but at the end, he was able to find the mobile phone. Another problem we discovered, this is we are making just one electrical stimulation in the same electrode with the same parameters. And you can see that, okay, we make one electrical stimulation and we ask, where is the perception? I say, here, and now it's here, and now it's here. What's happening? Because the electrode is in the same place. what's happening is that he's gazing to the last location, and the new perception is always down to the left, down to the left. And that means that we have to take into account the gaze when we are using a cortical device, and this is very important. You can see here what happened with Max. It's able to find something, but not in the right location. In the case of Max, the one problem is to find he has no eyes, but some cosmetic eyes. And you can see tenutosi in one of the eyes also. It was quite complex to measure the gaze position, but with the help of Avi Caspi, we managed, and we are able now to manage the location because the muscles are still there. It's able to move the prosthesis, the cosmetic prosthesis. And you can see when we ask Max to find where is the orange, it take also some time. Orange, and is able to take the orange without eyes, what is really amazing. But that means that we have to move. This is the standard way, the open loop stimulation. We have a visual scene, we process the data, and we send to the microelectrode array. And I think now we have to take into account also the eyes movements. And one important thing also in the brain is that in the brain there is feed forward and feedback connections. So there is connection going to the next place and connection coming back. And this dialogue is completely usual in the brain. And so we think in order to develop a usual visual sense, we have to emulate or we have to simulate this dialogue. We have to talk to the brain, but we have to also listen to the brain. And this is a recent paper by our group. We are showing, because our device allows to get recording, that from these recordings, for instance, we can have information from the threshold. Thresholds are the current, the intensity we have to provide. Just from the neural recording, we are able to make a very good prediction of the threshold. The correlation is 0.8. And the other one is a different experiment. We are changing the frequency and the brightness, and we ask for the frequency and brightness. And the last one is just the prediction, just from the neural recordings. Again, correlation is quite good. It's 0.8, almost 0.9. So we have another experiment. We are able to know if the patient is going to have a single perception or two perceptions just from the neural recordings. And I think this is very, very important for the future. Another problem, for instance, why it's important, the closed loop stimulation is neural habituation. So this is the first time we deliver one electrical pulse. There is some response of the neuron, but the second time is a little bit less, and the next time is a little bit less. That means that probably if we like to keep the perception, we have to take this into account. So now we are working with this. This happened. We have experiment. You can see that with the patient, it's the same. After some time, the perception is fading out. And we are trying to better understand what is going on. We are collaborating now with Pieter Rosma, and we are also making similar experiments in non-primate monkeys. Yeah, with similar experiments, the results are very, very similar, but in monkeys we can make many, many more experiments. And for instance, now we are just exploring different strategies. And we know, for instance, that in order to keep the perception, we can use, for instance, alternate electrode stimulation, now this and then this, or we can change the frequency of the stimulation. So, I mean, different strategies just to keep the perception. But the problem is very, very complex. So now we are collaborating with Michael Bejeler at the University of California, Santa Barbara, and Ciechi at the EPFL in Switzerland here. And so we are using AI techniques just to try to improve the way we are making the electrical stimulation. The standard wave is, for instance, if we would like to simulate one line like this, we stimulate the electrode like this, but this is not working. So now the idea is just the opposite. We know which electrical stimulation pattern induce this perception, and we are trying to stimulate this electrical stimulation pattern, and we have very, very good results using this. You can see here, because I mean, even from day to day, the response of the neurons changes. but from the results, from the models we are having from the AI are very, very good to make the prediction of what is going to happen. That means that I think that we have to move from the standard open loop stimulation to a closed loop stimulation, taking into account the eyes movement, but also all the brain signal that we don't fully understand yet. So we still need more knowledge, but I think for the future it will be important to better understand all of this. And I would like to show you also some results using this kind of approach. This is a standard experiment. So we are gazing, you can see that in this case the task was to find the small square in the screen, so there is no stimulation, and when the stimulation is on the square, there is the neurons, the electrical stimulation, and how the patient used this information. In this, the task was to find to find the cup. Is even faster than me. He was able to find the banana. And now he's able to find the spoon and put on the cup. So he's able, no vision at all, but he's able to perform some simple tasks. And in this case, we have for mobility, before for safety reasons, we are using this artificial environment. So here the task is to avoid obstacles. It's like a game. So for just to learning, we are using gaming. So it's very fun, and when he made some mistakes, say, oh no, try again, I'm going to start again, because I'm very good on this task. And his Max, also, no eyes, but he's able to find that there is something, and he's moving. What for us, the task is very, very simple, but for us it's really, really amazing. We also have in the lab one street lab, and it's an environment for studies of orientation and mobility. We also have an apartment, a full apartment with different kind of obstacles, and it's not only for our approach. So the idea is to use for people working in low vision, different drugs, and so on. And it's full of cameras, and so we can have different kind of designs. And we can add obstacles, we can remove obstacles. So we are very happy with the result, because we are using this infrastructure. We also have this is a corridor, again, with different kind of obstacles, and we can follow the patient using this facility. You can see here, Max loves to play football, soccer, and in this case, the task was to find the ball with our eyes, and then has to find the person, and is able to perform the tasks. And this is too simple, too easy, and we were making even every time a little bit more complicated, and now the ball is in different parts, and it takes some time, but after he's able to find the ball, as you can see, and he was not so sure, okay, and now has to find the portero, and send the ball, and this is in an even larger area. I remember Max was in the lab with several friends, and they were playing football with friends. And here is just the first time with Xavier. So he's a little bit insecure with the hands, but he's able to find it just, okay, say, okay. There is something in front. There is something, so move a little bit. And Dr. Livingston, I guess. So he's able to find that it's a human, and this is after some training. He's at the university, so he's able to move in the corridor, just avoiding obstacles. Again, it's a very simple task, but he's able to use just the pattern, the electrical stimulation to avoid the obstacles, to find the path. So we think that these experiments are very preliminary, but they are encouraging in terms of the feasibility of a cortical visual neuroprosthesis. In our case, for instance, we have all the blind volunteers have been explanted, and we have no fibrosis at all. So we think that the fibrosis is also related with the surgical technique, and we are using right now a surgical robot for our implantation. And these experiments, also very preliminary, shows that the UTAS electrode array allows the stimulation over a period of 6 months. Thresholds are stable. Pattern stimulation is able to induce pattern perceptions, and a bidirectional link with the brain is visible. And blind individuals can use these perceptions for orientation, for mobility, for daily life activities, and we can explain safely. But so I think we try to play the piano, and we are still banging the piano, because this is not the best kind of electrical stimulation. We are not releasing neurotransmitters, so we still need more knowledge, and there are still many open questions in the field. For instance, there is a limited number of electrodes. We are using just 100, and each eye is sending more than 1 million axons to the brain. So we need a wireless communication. So the neural interface is far away for mimicking the real anatomical and physiological connection. We still need a lot of information, from coding strategies, all the technological challenges, all the biocompatibility, long-term biocompatibility issues, and we need a better understanding of brain plasticity. We have seen today morning that retinal plasticity is very important, but brain plasticity is also very important, and probably we have to take into consideration both. And we have been working, in the lab we are working on also on plasticity. This is a recent study by our group, and we show, for instance, that electrical stimulation changed the brain. So the connection, the functional connection due to the electrical stimulation is changing the brain, and we also have to take this into account, and I think this is my last slide. Okay, probably you know this is from the City of Light. In this case, you remember probably the story. Charlotte, we call Charlotte, I think in other countries is the Trump, is in love with she was completely blind at the beginning of the movie, and Charlotte, the Trump, managed to get money to pay to the medical doctor to cure the blindness of this person, and she's unable to recognize Charlotte until she touched him, because she learned to meet Charlotte when she was completely blind, and she meets by the touch. So sometimes even if we are able to recover the vision, patient may be not even able to recognize people and so on, because they learn from other sensory modality. So the whole history is very complex, and I think we have to take all of this into account for the future. You can see now, but only when it's touching the Charlotte is able to see. And yes, well, this is all the people working in our lab. I would like to thank all the people working, all our founders, and of course all the blind volunteers that are helping us in a completely generous way. The only goal for these blind volunteers is to help others in the future, which I think is very, very, very remarkable. So we are very thankful to all the patients for all the help. And thank you very much for your attention. Grazie mille.
Con il professor Fernández abbiamo fatto un viaggio dalla fantascienza alla realtà, perché i pazienti operati esistono realmente, primo. Secondo, Eduardo, perdonami che qualcuno si farà i calcoli sulla tua età, ma Eduardo Fernández lavora da 40 anni in questo settore. Ha lavorato per tanti anni con il professor Norman negli Stati Uniti, ha sviluppato una tecnologia personale, ha sviluppato un laboratorio che non fa soltanto visione artificiale, ma fa tante altre cose, dalla optogenetica, gene therapy ed altro. E io ringrazio molto Edoardo perché mi ha fatto partecipare al suo progetto, e il suo progetto, ma il sottoscritto Andrea Cusumano ha avuto il permesso di parteciparci. Nel luglio 2024, il 15 luglio, ho partecipato all'espianto del chip corticale dal soldato ucraino che per colpa di una sciagurata granata di questa sciaguratissima guerra russo-ucraina ha perso completamente gli occhi. E noi rivedremo Max, che è stato così generoso di partecipare insieme agli altri 3 soggetti al progetto di ricerca, ma sapendo che avrebbe avuto rimosso l'impianto e che l'avrebbe avuto probabilmente, ma non certamente, rimesso in una fase successiva quando la qualità sarebbe stata migliorata. Quindi Edoardo ti ringrazio molto, e un'altra cosa, posso fare un annuncio visto che ci siamo parlati questa mattina. La Macula Genoma Foundation parteciperà l'anno prossimo, perché ogni 2 anni c'è uno straordinario incontro che è organizzato da Edoardo dalla sua università ad Elche Alicante, che si chiama The Brain and the Chip, e il prossimo anno la Macula Genoma Foundation parteciperà in maniera attiva a questo meeting perché è un'eccellenza internazionale. Quindi ti ringrazio molto per quello che hai fatto, per il futuro reso realtà, e penso che molti dei partecipanti oggi, molti dei pazienti che hanno perso la visione non potranno riaverla con un microchip retinico di Daniel, perché hanno problemi di nervo ottico, perché hanno avuto dei traumi, potranno riaverla un certo tipo di visione, speriamo presto, quando la tecnologia permetterà di passare dai cavi al wireless, a risolvere dei problemi anche di batteria, di funzionalità, ma non credo ci vorranno molti anni, specialmente se la velocità con cui tu procedi e il tuo gruppo sarà questa qua. Quindi grazie ancora Edoardo.